have two people from the NIH here. We have Dr. Amanda Ombrello, who I believe many of you already know. She is the lead associate investigator for Dan Kastner's autoinflammatory disease protocol. And she was the first person who reviewed a chart for the Undiagnosed Diseases program. And that was a really important moment for all of us in this room. And she was the one who put two and two together and has been working on our behalf ever since. And this is Natalie Deuitch, who knows all of us very, very well because she works in the lab and does diagnosis and genetic testing. So she knows more about us than a lot of other people do.
Knows your blood, maybe not your face, but
it's good to put numbers and faces together.
Mutations in faces.
Yep.
So they are going to talk to us about what we know and how we're moving forward with understanding the disease. And they are willing to take questions all the way through, so if you want to stop them and ask a question, you can. They'll be watching for questions. I'll also stay up here so you can flag me down if we're not noticing you. And we have a whole hour, so let's go. Thank you so much for being here, both of you.
well, we're excited. We have some slides. They'll come up here in a second. But I hope you guys have been having a good day. We come from the land down under, and and it's a good time down there too. Probably not as fun as here. But like Catherine said, we're here to kind of give you an overview of what we know and maybe where we're heading with things. And like she said, any questions, anytime. So briefly, who are you?
Okay, so I am, I have a bachelor's of arts in biology. So I work down in the lab. I'm a post-bac, so I'm actually in the process of applying for genetic counseling. And you guys honestly have really inspired me that that's the direction I want to go. I want to stay in genetics and work with patients with diseases like all of y'all. But
when you send in blood, she gets it.
Yes.
She processes it. She does the genetic sequencing. She does the ADA2 levels. She does that.
I am the vampire that makes you give me all of your blood all the time.
Yes, yes, yes. Such a nice smiling face.
Yes.
I am Amanda Ombrello. I hail from the Midwest, Missouri, and I went to a small school in Springfield, Missouri, down by Arkansas area, and went to medical school at St. Louis University, and then I did my residency and fellowship there in internal medicine and pediatrics and adult and pediatric rheumatology. And then my husband decided it was really a great idea that we were going to move out to the East Coast because he really wanted to work for this guy named Dan Kastner. He's an adult mom. My husband's adult and pediatric rheumatology too. And he's like, I need to do my best to get it going with research. Come on, let's move out there. So about 7.5 years ago now, he put me in contact with Dr. Kastner, who I like to say we fell into each other's laps because as Dr. Kastner was rising up the administrative, executive level, he needed somebody who could run his clinic. And as I had both adult and pediatric training, I was perfect, and I needed a job. So out I came, and I've been working with him in the group ever since. I am a mother of 3 kids. That's my Olivia. She's a first grader. And the middle one there is Jim. He's James. He's 4. And then little Mr. Smiley is Evan. He's 2. And the reason I put them up there is I don't want to bore you with cute kid pictures because you guys all have cute kids. I know that. But I wanted to. Some of you know this, but I don't think a lot of you do. That's my 4-year-old, my Jim, and that's when he was a little smiley baby. And Jim, so let's see, he was born in 2012. I've been working here for 2+ years, and he was born, and at about 2 months of age, he had a fever, and my husband and I should have taken him to the doctor, but we said, no, he's got an older sister, I'm sure that's what it is, brush it off. We're pediatricians, we know. And so then we started sending him to daycare, and at almost 5 months of age, he started having these fevers, and they would go up to 105 and last about a week. And this was going on month after month after month, and he gets sent home from daycare, and we have no other family out on the East Coast, so my husband and I would have to come home and stay with him, and he'd start crying overnight 2 days before these fevers would come, and it took us a while to find out that pattern, and we'd always be frustrated, like, why is this kid crying all night? I'm never going to get good sleep. But finally I started saying, no one in his daycare class is sick. My daughter's not sick. The doctors just keep telling me this is a virus, but he's never had an infection and needed antibiotics. Something is wrong. But I said, I'm just overcalling this because I do this. I see people with fevers, so there's no way. And at 1 year of age, he had a lead test drawn, and they did a blood count at the same time, and his platelet count was 700-something thousand, and his blood count, he was profoundly anemic. And so finally I said, okay, I'm not looking the other way anymore, and there's something wrong with my kid. And it turns out that, maybe you'd say miracle of all miracles, my Jim has an autoinflammatory disease and actually gets seen at the NIH by Dr. Kastner. And so I just put pictures of, not because he's cute, but, he's had a lot of different things. And he's had a lot of eye inflammation. And just the top picture there is him just this week without external eye inflammation. He gets a lot of throat and sores and lymph nodes and joint issues. But he's doing okay. He's doing well. But I just put this all out there for you because I want you to know that not only do I treat and take care of many of you guys and your children, but I kind of feel like I understand from a parent level too how it's scary to not know, because we don't know what he has. We've had sequencing, and so far it hasn't come up with anything. So I get it. I know what it's like to kind of not know what tomorrow's going to bring, and to hope. So that's all. Anyway, why we're here though today is this wonderful, wonderful disease, deficiency of adenosine deaminase type 2. And let me give you a little background here, because I think this is an awesome story, and I hope you guys think so too. And so well before I came to the NIH, in 2004, Dr. Kastner had a 2-year-old little girl who got referred to the clinic to think maybe she had a disease called Muckle-Wells because there was brain involvement. Well, there was no way that she had Muckle-Wells because she had a history of 2 strokes, one in the spinal cord and one in the internal capsule, and she had a history of fever and rash and went on examination. Those are actual pictures from that time. You can see her rash, and you can see that cute little sad face that we were probably torturing her. And they did the testing that was available at that time, and nothing was revealed, but Dr. Kastner, gave it his best, as we often try to do, and put her on anakinra, the IL-1 blocker. And years went by, and she had been, affected from her strokes, but she didn't have another event, and her fevers were gone, and so she was going along, and we didn't hear a lot from her, but she followed up yearly. And then in 2011, I had, because I reviewed charts for the Undiagnosed Diseases program at the NIH too, and one appeared on my desk, and it was a referral from Texas about this little girl who had, 6 strokes, and where they had been, and she had a lot of fevers and rash and anemia, and tried all these medicines. And so what I get to do as a reviewer is I get to say, yay, this is a good one for the UDP, or no, it's not, or I want it. And I said, I want this one, and bring her to our team. So she came out to see us in the fall of 2011, and cute as a button. And then the last part of the story, and this is so cool that I still had this, in January 6, 2012, one of the other attendees on the team, Dr. Carol Barron, sent an email, and so you can't see it, but she's like a little weekend reading, and it's this email from this doctor, Dr. Sheetal Vora, and she's talking about a little boy, 6-year-old, who had 5 strokes and fever and a rash. And I did my little weekend reading as I was supposed to, and I emailed back on Monday, and I said, Carol, This patient sounds a lot like, or this little case sounds a lot like patient H. Luton, who has very similar clinical characteristics, fever, strokes, rash. We are going to be doing exome sequencing on her, and I think this would be another great case to add at the same time. And so we had then 3 cases that we sent for, because we brought this patient, and we sent this sequencing out, and lo and behold, that is where, oh, look there he is. There we have our sequencing results. And, you know, 1 is the first little girl, 2 is the second, and 3rd is that adorable little boy. And as you can see, they all shared mutations in the same gene, although the little boy decided to be a little difficult and have a hidden one. And the lab came to me, you weren't there, so you weren't responsible, and said, nope, he's only got 1. He can't have this. And I said, oh, no. He has this. You have to go back and look because he's a match. They're a match. He has the same thing. And so they worked really hard and figured it out. So from that point, we now have 25 patients that we actively see at the NIH, and 17 of those have had strokes. And over time, they've had 58 total strokes. So this doesn't happen once, it happens multiple times. And they tend to have their first stroke, the median, so middle age of onset was just over 4 years of age, and over half presented before 5 years of age, and the oldest in our cohort had his first stroke at 12. And they're generally in the same areas, and you don't need to know where these all are, but you may have heard these buzzwords over time. There's brainstem and thalamus and internal capsule and basal ganglia. And if you line them up, they could be in the same place over and over and over again. So those are just some of the pictures of some of our patients' brains, and you can see I've highlighted them for you. And then we've also seen that there's these hemorrhagic strokes, which I know some people have experienced firsthand in this room. And 4 out of our 17 have had the strokes, hemorrhagic strokes, and 2 have had multiple, so there's been 6 total events. All of ours were on some form of anticoagulation at the time, so like aspirin or something else. But there are patients out there who haven't seen us, who were on absolutely nothing, who have had these happen too. So that is definitely something that I recommend patients avoid. You know, even a baby aspirin, I think it just, there's already a risk in this disease, and so we tend to say, you know, please try to avoid, because we just don't want this to happen, because sometimes this can have more after effects than what any number of the other strokes have had. Okay, so this is a pretty picture, and what it shows is what we kind of know right now. We don't have it all figured out, but we know that if you have normal levels of ADA2, what it does is it's a growth factor for endothelial cells. So your blood vessel will look pretty and smooth, but when you have low levels, it's got this disruption in the endothelial cells. So you can see that is not a pretty and smooth blood vessel that you see cut in half there. It's kind of like jagged looking. And then on the other side of the coin, when you have ADA2, it helps to promote this type of cell called an anti-inflammatory macrophage to develop. But when it's low, those anti-inflammatory ones cannot develop. Therefore, the inflammatory ones take over. There's more of them than there are the anti-inflammatory ones. So you've got these inflammatory macrophages which release TNF-alpha, which is important when it comes to treatment, and you've got these jagged vessels, and when they all come together at the same party, that's when you get the problems. You get the fever, the rash, the strokes, the pan or other vasculitis. This is what we believe is the predominant presumed mechanism of disease. So I'm going to let Natalie talk for right now, because she's going to tell you a little bit about what she does.
Okay, so as y'all know, this disease starts with a gene, this gene CECR1. I wish it was called the ADA2 gene. That would make my life much easier, but it isn't. So the CECR1 gene acts as sort of an instruction manual. You know, all of your genes in your genome are an instruction manual for your body and tells it how to build proteins and all of these building blocks that make you, you. So CECR1 makes this protein, ADA2. That's what it looks like. It's way too complicated looking. And then this protein, ADA2, does all those things that Dr. Ombrello was just talking about. So it does all of this stuff. And you can imagine how if your gene maybe looks a little bit different, if that instruction manual is spelled kind of wrong, this protein might look a little bit wrong. It might be the wrong shape, or it might interact differently. And that's going to affect all of these things downstream. So you've probably heard that DATA2 is recessively inherited. So all of the genes in your body, you get half of them from mom and half of them from dad. You get 2 copies of every gene. So you have 2 copies of the CECR1 gene. And why it works that way, if you can see this graph, is that if you have 2 copies of perfectly normal CECR1 or ADA2, you're functioning, your enzymes are working at 100%. So if you have 1 copy, maybe from mom, that isn't working so great, but the copy you get from dad is good, your enzyme isn't going to work quite as well, but it's still going to be okay. So your body actually makes more of this protein than you really need. Just as, you know, a precaution, there are a lot of things that go wrong. So we don't really see any symptoms if you just have 1 copy of this gene. But where trouble can come about is if you have 2 copies that don't work quite right. If that gene is spelled a little bit wrong and the body can't tell how to make that ADA2 protein. And that's when you see DATA2. So you have to get a copy from mom and from dad. The way that works sort of downstream when you have kids is that, you know, if you get 1 copy from mom has 1 good copy and 1 bad copy, dad has 1 good copy and 1 bad copy, each child has a 25% chance of getting 2 bad copies. So, you know, ideally if you have 4 kids, 1 will be affected, 2 will be carriers, the next 1 will be totally normal. But it doesn't always work that way, and we actually have a lot of families where there'll be, you know, 3 kids who all inherited the disease. So I want to show you guys a little bit about what it looks like when your blood samples come to the lab, so when I take all that blood. So first what I do is I extract your DNA. So I get rid of all those cells and all that stuff and just pull out that genetic instruction manual. Then I run it through this machine, and it spits out all of these letters. So the CECR1 gene has thousands and thousands of different letters, and each letter tells the body how to build that protein a little bit more. It's just like words in a book. So then I sit on my computer for hours and read all of these different letters that make up your gene. So one thing that I think is important to point out is not every misspelling in your gene does something bad. There are lots of different ways that our genomes are spelled differently. And it's just like words. So most of you guys probably spell the word color, C-O-L-O-R. My family's actually British, so I spell it C-O-L-O-U-R. If you read that, it would look kind of different, but you could get the gist, right? You'd know, all right, she's talking about colors. But sometimes that instruction manual is spelled, maybe it says cat, or maybe it's just gibberish, or maybe there's no word. And that's when the body says, oh, I don't know how to make my ADA2 protein. And that's when you start to get problems. So there are 2 million different places in your whole genome, not just CECR1, where things are spelled a little bit differently. And in the CECR1 gene alone, there are 302 different places where there are different spellings, but not all of them are bad. So this one down at the bottom here, this piece of CECR1 gene, this is just like a total normal person. That might even be my DNA. But there are 2 places where it's spelled differently, but it's not a bad thing. And on the side here, this is just a list of all of these mutations in a big database of people who don't have DATA2. But what I have to do is I have to pick out which mutation is going to say spell cat instead of color and is going to cause disease. So we do that in a couple different ways. So this is somebody's DNA where they have a misspelling. First, I can look at the protein, like I showed you, and see, you know, okay, well, if I change this letter to this letter, what's it going to look like? We can also look, it's kind of cool, but you can look at this gene and see how it's spelled in all these different animals. So you've got humans, cats, chimpanzees, dogs, and for the most part, they all spell this gene pretty similarly. And this is kind of cool going forward to look at the zebrafish. Some of you have heard about this. So we actually made a data 2 zebrafish where we, messed up the gene and saw what happened. And I say this because the zebrafish and humans share 85% of their genome. You are that similar to a zebrafish. But when we mutated this gene in the zebrafish, we saw strokes. We saw symptoms kind of similar to what some of the patients have. So then the last way that I'll look and see if a misspelling is really significant and is really going to make the wrong protein is I'll look at the serum levels. And I know a lot of you guys have had this done recently, and it's something that we're starting to do for all of our patients. So what you can see here, the pink ones, those are controls. So those are normal people who have 2 copies of the CCR1 gene, and they're functioning at 100%. And then you can go down and you can see carriers. You can actually see, so kids have more ADA2 protein than adults, and that's because it works in development of those blood cells. So you guys are growing and building all these new things. And then the patients are down here at the bottom. And you can see, so these are all of your different genotypes lined up, and you can see sort of the different variations. So these people, I don't know if this clicker works, but the low ones are all of our ADA2 patients. And you can see they're all kind of much lower than the carriers and the controls. And it's also important to kind of look and see that there's some variation even between the patients. Everybody has a slightly different level. And hopefully in the future, as we get more and more patients, we can say, oh, look, this G47R mutation makes people's level lower than maybe another mutation. And it'll help us say which mutation makes things bad and which things make it, not so bad. So through sequencing all of your genes, and why I'm talking so much about all of these misspellings, is we've been able to find all of these different ways that the gene can be misspelled. And, mutations are mutations, you guys have the disease, but by kind of targeting in on these things, it'll make it easier for other researchers to be like, oh, here's a patient with the P425A mutation. Well, Emma at the NIH also had that mutation. That's definitely it. That's definitely the reason why the disease is the way it is. And then another thing that we've been doing, so I went through a database of people who've had genetic testing of the CCR1 gene, not because they had data 2, just because it was getting tested with a lot of other genes. And we were able to look through at those mutations that are predicted to cause a disease and predict that 4 in 100,000 people, every 100,000 people, have a combination of 2 mutations that could cause this disease, right? That's pretty crazy. That's a lot more people than we would expect. So sort of moving forward, we want to see exactly which of those people actually have the disease and which maybe don't, and that'll help us narrow down which mutations do and don't affect it. I guess sort of in the context, we're really trying to figure out, I know some of the patients have PAN, some have strokes. What about the genome causes all of these different phenotypes or different clinical outsets? So is it the different mutations like I was talking about it? Is it the G47A or this P425, all these different letters? Or is it other genes in the genome that kind of work together with the CCR1 gene? Or is there some environmental trigger? And we really don't know, but we're learning, and you guys are definitely helping us tease out this very complicated thing.
Because we'll see that even within a family, for instance, in one family where we have 3, one of them has had 2 strokes, one has had more of a fever component, and one of them has had almost nothing. But yet they all have the same mutation. So how do you describe that and reconcile that? But that's common for what we see in genetic diseases, is they're not carbon copies from one another. You get this variability. So just because it was one way in one patient doesn't mean it has to be that way in another. So here are some pictures of some of the other clinical manifestations we can see. We have some very prominent livido there in one of our patients. As you can see, it's quite profound. And then we have a very handsome hand that we're looking at there. And this patient actually has severe Raynaud's type of clinical presentation, where in the cold he gets purple and white coloration of his fingers. But it's not just your ordinary Raynaud's, in so much that I call this the ghost hand, because normal hands that aren't affected, you're going to see the ends of the fingers, but his are kind of fading out. And that's because his blood vessels at the tips of his fingers have been so affected by the disease that there's very little blood flow getting there. And so we're doing our best to counter this, because this is how he came to us. So this was already pretty severe by that time. We see a variety of eye findings. Some are a result of stroke, some maybe not, because they present at different times and there's no lesion that occurs with it. And then the leg on the right, it's kind of hard to see in here, but there's more of the nodular component that you see with the polyarteritis nodosa or pan nodules compared to just the livido. We know that there's varying forms of immunodeficiencies too, so a lot of patients need IVIG or have an infection history. Some patients have more severe immune deficiencies that we're hearing about downstairs. And then there's also a lot of different blood disorders one can see. Like some patients don't make any red blood cells really at all. Some just have more of an anemia. Some have complete bone marrow failure, so that means white blood cells, red blood cells, platelets, and once again, we don't know why the differences are there, but they are. There is also a subset of patients, and some of you are in this room, who've had some more of the GI, meaning stomach and intestinal disease, that sometimes decreases the blood flow to your intestines and requires you to have that disease portion taken out, and then kidney disease where you've infarcted or cut off the blood flow to your kidneys. So there are just any number of different ways this disease can present, and I think what we're learning to do is to keep an open mind. And I don't think, it's very rare that we turn down somebody's request for a test. If a doctor contacts us and has some sort of suspicion, we generally say, sure, we'd be happy to test, because at this point, we just don't know how broad this disease is. So we use the anti-TNF medications, and some of you may wonder why. So in our cohort of patients who have had strokes, I went back and looked at all the medications that have been tried prior to going on the anti-TNF. And it seemed there was a lot of patients who were on steroids or prednisone, and then Cytoxan or cyclophosphamide, and then there were the IL-1 blockers like Kineret or anakinra, and IVIG, and mycophenolate. I mean, some of these are probably familiar to you, but they're heavy hitters. They're all heavy hitters, and a lot of them can be used to treat, inflammation like fevers or brain. Cyclophosphamide's really big in treating brain vasculitis. So these were not bad medications to choose, but what we see when we look back on our patients is that you could have put them on all the prednisone in the world, but it wouldn't stop them from stroking if they were going to stroke. It would make them feel better if they were having fevers. It would make, sometimes other joint aches go away. But if they were going to stroke, gosh darn it, they were going to stroke on top of all the steroids in the world. And the same holds true for all the other medications, is that they might kind of work, but at the same time, they weren't stopping these strokes from happening. So that's a steep uphill to face when you start seeing these patients, is they've had, the whole kitchen sink, everything but the kitchen sink thrown at them. So what do we do? And then also, we looked and saw what kind of anticoagulation medications they'd been tried on, and a lot had been on aspirin or enoxaparin or Lovenox or Plavix, which is clopidogrel, warfarin, or Coumadin. You can see them all listed there. A lot of people have been tried on a lot of different things. Okay, so this is a little bit confusing to look at, but first look at the two bottom, the C and F, and you can see it's a nice cool blue. There's no red on there. And that is a normal person's blood vessel when we're staining. That's what normal looks like. Now, the red on the right especially is indicative of TNF, or tumor necrosis factor. So these are data 2 patients in the top 2 panels. And you can see on staining around their blood vessels, there's red. That means there's TNF there. It's surrounding the blood vessels. It's in some of the blood vessels. So we said, that might be something we can go for. And none of the patients had really been tried on that, aside from one, actually, who'd done quite well, but we didn't see him till later on. And we also talked to some colleagues over in Israel who had more of a pan group, and they'd had a few of their patients with the intestinal problems who'd done very well on some of the anti-TNFs, because they were just discovering data 2 at the same time as we were. We published it in the same journal the same week. So what we started doing in June of 2013 is starting our patients on the anti-TNF drugs. Now, there's a lot of different ones to choose from. We tend to use Ombrello or etanercept as a good starting point because it's very easy to dose in young children because you can do it based upon their body weight. And so the vast majority are started on that, and they do very, very well. There's some patients who we found have a little bit of refractory or breakthrough disease on Ombrello, even if we've up-titrated the dose to a good level. So we switch them to other anti-TNF drugs. And this is a little confusing for why we might be choosing this, but I'm going to try to explain it. I think it's in the next slide. Hold on. Oh, well, hold that thought. There's a slide on it later, and I'll try to show you why we think that the switch might be better. So this is going to be a little bit mathy, but earlier this year, so it had been about 3 years since we started the first patients on the anti-TNF drugs, and I said, okay, let's go back and see how we're doing. And so when you're figuring something like this out, you have to choose a time 0, meaning that's where the time clock started. And so I looked at it using time 0 as the time of the first stroke for the patients. And then I added up all of their months that have gone by for each patient and then added it collectively together to get a grand total of 1,173 patient months went by using that time 0 as a time of first stroke for each patient. During those 1,000 plus months, there were 55 strokes that occurred. And so that was happening like every 15 months or so. So it was happening regularly. So after we started the TNF meds, using that then as like a new time 0, we went forward and counted the number of months and added them all together. And there were 370-something months. And during that time, we saw absolutely no strokes, which is beyond amazing, almost too good to be true, because if you would have kind of looked at the trajectory of the prior one, we should have seen over 10 strokes in that amount of time. So the fact that we saw none with the numbers at the bottom, the 10 to the -8, that is so very, very statistically significant that this is being written up to be published, because we don't want another child or another teenager or another adult to get told, no, your insurance company is not going to approve this, and then have to wait to get a treatment that could be completely life-altering for them. And so that's my goal out of this paper, would be to make it, because there's nothing more nerve-wracking for us than to say, okay, we've got a new patient, and of course you want to bring them to the NIH the next day, but it just doesn't work like that. Life isn't so easy, and because we can give some medication to tide them over until the insurance company sees the light of day, which we argue very well. But you kind of really have that fear now that we see that this has happened, that you don't want another day to go by or another stroke to happen. I mean, that's the weight of the world right there on your shoulders. And just to prove that we were right in what we were seeing, is we did it another way, where if you use a different measurement, because people could say, you use time 0 as a time of first stroke. Well, you are artificially inflating your numbers, because what about all that time that happened before the stroke? You should have figured that in as well. So instead, you use time 0 as the time when you start the drug, and then you know how many months have gone, patient months went forward, and then you go that same amount of time backwards. So we had 328 patient months on treatment, and then when we went back, there were 30 strokes that occurred across our patients in that time. And then when we went forward, there were none. So once again, it's not, you can't really argue something like this. And of course, in a perfect world, things would be perfect. And I'm an optimistic person, but I also feel that, there's going to be something happen at some time. The likelihood that this will remain 0 forever is probably not the case. There'll be breakthrough strokes, but this is just such a game changer, I think, for this disease, because other things that were being looked at, our bone marrow transplant, which has been extremely effective in some of the patients, and especially if they've had some of the bone marrow failure, or the sort of with the red cells where they're not making any. It can be very, very good, but it doesn't come without its own set of risks that can be had. Like, people can get what's called graft versus host disease, or they can die during the whole process because you wipe out bone marrow to replace it with new. And then, unfortunately, there have been a case or two now that I've heard of where afterwards the patients went on to develop vasculitis. So although a cure for many, just like I say, 0 won't happen forever, bone marrow transplant won't be a cure for all, if that makes sense. So also on medications, we see dramatic changes in labs. The inflammatory markers, sed rate and CRP, kind of shoot down. And the platelet counts, if they were elevated, kind of come back down to a more normal level. And then the iron levels and the anemia tends to correct as well. So not just stroke response we're seeing lab response. And then this is a really cool slide, I think. All right, so if you don't believe the numbers and you don't believe the stats, here is something I don't think that any of you can disagree with. This is at time of starting TNF in one of our patients, and you can see that there was a lot of livido. There was a very prominent belly because the spleen was really, really big, because some of the patients can have liver involvement. And she was requiring steroids at pretty high dose, and she was small, at less than the 3rd percentile for her height. And so that was June of 2013. 25 months later, this kid grew over 20 centimeters. That's like a ton. Her prednisone dose went to 0. She, as you can see there, I mean, you're looking at a different kid. The spleen, although still large, is not getting larger. So you kind of grow with 20 centimeters more into that spleen. And the livido isn't quite so prominent. And the secondary effects of the steroids that many people saw aren't near as prominent either. So that is so cool. As one of the doctors who sees a lot of these patients, is you get to see that transformation happen. And it's not just that patient. We've seen it in many, many who have been, so ravaged from the disease, and then also from the medications they've been put on, have this complete transformation. Okay, so this was the slide I was looking for earlier, and it was about why do we choose one over the other. And so Ombrello or etanercept, so in the middle, that's what an antibody looks like in the body. And antibodies, we typically produce antibodies to infections, because that's an antigen. is the infection. It can have an antigen, and they want to bind it and get rid of it. Okay, so that's what in all of our bodies, that's what happens. So when tumor necrosis factor is produced in excess, smart people about 20 years ago started making these drugs where they could form like an antibody that would bind to the TNF and then would make it not able to cause the inflammation. So Ombrello is there on the left, and as you can see, it doesn't quite look like the antibody in the middle. And it's because they kind of engineered it. It's called a fusion protein, and they took the curved purples are TNF receptors, so they actually work the same way. They bind the TNF, but it's not an antibody in the same way that there on the right you see Humira or adalimumab, and it looks very similar to the antibody in the middle. So Ombrello works, and it works great, but we question whether or not in some of those that get the breakthrough, if it's just because there reaches a limit that it can work if the disease is really wanting to be active. And so we're trying to switch people then over to the ones that look more like a real antibody. We call them the monoclonal antibodies. It's just a class, it's a big word. But that is why we're making the change, and that's our rationale for changing what we're changing. And some of you got changed just this week, and that is why, is we're trying to see if it works better. And so far, our clinical experience is, okay, she, a patient, had more nodules breaking through on Ombrello. Well, we switched her to Humira, and they cleared right up, and she's doing great. We've switched to other ones that look like Humira, like there's one that's called golimumab, and where we took a patient and switched it and got much better circulation in the extremities because of it. So we're seeing that it can pack an extra punch. So some of you patients and parents may be saying, how long do I need to stay on this medication? There's a risk that can come with medication, and maybe, maybe we could go off and things would be better. It's a good question. So here I made a couple of graphs over what some of our patients look like. So patient A, as you can see, had a really intense couple of years there where there were 7 strokes. Boom, 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 boom. Like, so life was not good at all. And every medication was being tossed at this patient, trying to get these strokes to stop. And it wasn't working. And so that's what we would call one of those intense inflammatory episodes where the disease was very, very active. And as you can see, the blue line is where we started Ombrello, and it was like it shut it off right there. And going forth, we've had the benefit of nothing else happening. Well, conversely, we have patient B. There he is. And he had 1 stroke out of the blue, and then went years with nothing, on nothing, and was doing well, but then had 2 strokes pretty quickly. And so it's a completely, what I'm getting at is it's a completely unpredictable disease. And inflammation doesn't always have to happen, because remember, steroids are going to reduce inflammation, but steroids don't stop strokes in this disease. So we don't know enough about it to say what's going to cause you to have an intense active part versus, years of pretty quiet. But we do know that these can happen at any time. So we say it's not worth the risk. Don't stop the medicine. Don't stop the medicine if you're sick, because it's very common for doctors to say, oh, you're sick, you better hold this immunosuppressant. And that's what you do in a lot of rheumatic conditions. This one, you should never hold it. Please don't hold it. If anyone ever tells you to hold it, call your rheumatologist, or if you come to see us, call us and let us tell you not to stop it. Because sometimes the disease activity revs up when you've got an infection. So that is absolutely the time you do not want to hold the medication. So please, don't stop. Oh, and I guess that's the end of the formal part of our presentation. You know, it takes, there's a bunch of us. We owe it all to all the people that we work with and all the researchers we have, and that's just the tip of the iceberg. There's hundreds more, as evidenced by the group that's downstairs. But we are happy to answer any questions or try to answer any questions that you have. Yes.
On the Enbrel, our son takes Enbrel. What would you see that would make you change to another one? I mean, how would you know?
So that's a good question. And I know for your son that, thank goodness, there hasn't been a whole lot clinically to go off of. He's had some Livido. And I don't know if that's changed at all for him on the Enbrel, if it's just as, because we see some patients, the Livido still lingers. But I would say sometimes it's something so subtle as energy levels decreasing or concentration in school for the younger kids. You know, something is off, something is changing, and or they complain a little more of joint aches and pains. You know, if they have a tendency to get nodular rash, you may see that. If they have some GI symptoms, they may start having more pain after they eat, especially. Some start getting low-grade fevers or lymph nodes. Like, these are all indicators that we need to think about either upping the dose, because also with Enbrel, there's a lot of ways you can go because kids are growing. So they're always growing out of their dose. So you always are having to readjust. So we look for that too. But you're right. When there's not a whole lot to go on, it might be hard to know. But make sure that we're getting up-to-date weights so that we can make sure he's maximized on treatment.
Can I speak to that? We just switched this week. I mean, would that be appropriate?
Yeah, yeah, that's great.
We just switched this week. Well, it's been about a month. We were on Enbrel for about 2 years with our daughter. She's 5, and she was having pain persistent pretty much the night before we would give her Enbrel. She started developing a lot of different nodules. At one point, the doctors were thinking maybe she had some other condition develop because she was getting red marks too. And throughout the summer, so going back a few months, we kept increasing it to try to keep Enbrel, the drug that worked for her. But it was just pretty clear over the past 2 weeks with the pain. I mean, she couldn't walk. The red marks, it was really awful. So we decided to make the switch, and then actually we made the switch, and it was 14 days, but on heavy dose of steroids and the Humira, that wasn't working either. And this week she couldn't walk, and we were in the hospital on Wednesday also. So now we're doing it every week. So even with the switch, you might find things, and just these doctors are amazing, and we are indebted to you and all that you do. So, but you'll know. I think you'll know, and I think all the parents that you know. So, thank you.
I saw another hand. Yes.
So, when you have the disease and it focuses more on the, like an absolute neutrophil count.
Yes.
He's been at 0 for a very long time. Neupogen started to help that, but then as the spleen got bigger, so then you had to stop the Neupogen, which they had to give them again so that the airplane ride and stuff like that would increase. When you have that case where it affects your cells and your immune system, how often does that cross over to the vascular problem? Does it, or does it kind of stay in one lane?
So that's a really good question. And I think I might have mentioned we haven't seen in our cohort at the NIH any where they had the complete wipeout of the bone marrow, but we've had a lot of patients who have, like, for instance, a milder immunodeficiency, or they'll have a period of time in their life where they are at the hematologist because their red cell counts dropped really low. So we have variations in the middle of, like, overlap. We get overlap, but we haven't seen, tons of strokes with no neutrophils, right? But I'm sure there's patients out there that do. We just haven't seen them yet, is my take home, because I'm hearing about these other cases downstairs too, and you'll hear about somebody who had bone marrow failure and there was a stroke too. So there is overlap at times.
So it could be that it could almost progress to that?
I guess
You just don't know.
no,
Like, there's just not enough.
there's not enough data. You know, a good question would be, okay, and I wonder about this too, if I've lived 20 years and I haven't had a stroke, is my risk for stroke still there? Am I going to be one of those stroke patients? And so far, like I said, our oldest one that we've seen was 12 when he had his first stroke. But those who stroke on and on and on. It doesn't stop at 20. It goes forward if it's going to. We don't know enough now to know, you know, if there's a point where you're in the clear. It's too soon to tell. And that's why we are so just like, I don't care if you haven't had a stroke, you still need to go on this medication because maybe we can prevent that.
And so do you have a certain number of, like, his bone marrow's at 40%?
Mhm.
Do they have a number of where the risk would be worth doing a bone marrow transplant, or is it case by case?
It's done case by case generally. And the ones that I've heard about, because it's not like it's a popular thing, but the ones that I've been hearing about had complete failure, where there's like nothing and they needed it. Or, you know, the person who had no red blood cells, they were completely transfusion dependent. All right, so red cells, getting red blood cells. And over time, when you do red cells, you get iron overload in your body too, a whole different complication. So that deposits in the liver and can cause liver failure from a different reason. So when you've got that as your future, that too has prompted some people to have transplants. So it's case by case. I saw a hand. Yes?
You had mentioned that the gene is needed more for children because growth and development is used in that. Other than stroke, have you seen the effects lessen with age as they get older and they don't necessarily need it as much for growth and development?
I think we have one patient in this room in particular who can attest to the fact that the disease doesn't necessarily lessen with age. Once again, it can kind of be case by case, and as you saw in those graphs, you can have periods of time, but we have one who had her most severe stroke in her 20s, and we've had other ones that we know about too, where they've been in their 30s and had a really bad one. So I wish I could say it did decrease with age. That would be awesome. But I don't want to paint that because I don't think that that's true for a lot of patients.
Were they on medication?
No. I look at those, I say those patients were born too soon. That's how I look at it. You were just born too soon. Hopefully the patients now are being born at the right time, okay? That's the way we look at things, is that, and that born too soon, but they're here now, so hopefully that too, no matter what happened in the past, we are altering what is going forward into the future. We are positive, okay? Yes?
I lost some vacation, and I saw there that Ombrello doesn't take the disease away, but it only suppresses it. I had vasculitis on my legs, and yeah.
That's the correct way of saying it, is that Ombrello's not a cure, but it's a treatment. All right, we're looking for cures, and sometimes like a bone marrow transplant can offer that if they do it well. There's smart people working at researching replacement forms of ADA2, but that takes a long time to develop because it has to be safe and it has to be conducted, you know, lots of research on it. But that also would be a goal, that perhaps if we could make a form to give, that if we replace it to a level of what the parents have, which is not high, but enough that we could combat the disease that way too. Yes?
Is there anything with diet or anything like that that could help or?
So we haven't seen a direct correlation of anything with diet. Of course, we tell all of our patients to try to live healthy, right? But with all of our autoinflammatory diseases, it's not that that burger is going to be the one that does it, or if you're gluten-free or whatever, dairy-free. None of that really seems to matter. It's going to happen if it's going to happen. Yes.
And this medicine, the Inaugural, you don't hear nothing about making the same kind of medicine, but there's not being necessary to do the injection, like the same thing, being in a pill or being in a liquid.
I know people are working on that, right? Because goodness gracious, the vast majority of all my patients would think I was really cool if, instead of looking at me with daggers in their eyes when they're 5, 6, 7 years old, if I could say you don't need a shot anymore. And they're working on it. It's just not there yet. Because like I said, these things take years of testing and development to get to the point where FDA can approve them for use. Yes?
Is there a difference in the disease when both parents are the same carrier and parents that the different kinds of code on the DNA?
Not that we've seen especially yet. And that's one of those things that we're really starting to kind of figure out. As we get more and more patients, we'll be able to say, you know, oh, people who have the same gene or same mutation look the same. But, you know, there are only 25 people that we see. There are, I think, about 250 diagnosed worldwide. So that's just not enough to really know yet. But we will. I promise.
I'm so sorry. I forgot to increase my last slide because I wanted to show you that, like I said, now is a good time. These are many of the patients in our NIH cohort, and I've told you the data. You know they've had problems. You know there's been issues. But these are recent pictures, and every single one of them has a smile on their face and looks really darn cute. I have time for a couple more questions, but then I actually have to run back down and be on another panel that starts at 12:10. So it's not out of disrespect. If you want to write your questions down or
you can text them.
Text them, yes, text them. And I'll be around later. If there's something you think of, I have email capabilities. I mean, I think I'm pretty approachable and pretty easy to find. So.
Just a quick question.
Yeah.
Does nationality have anything to do with what you found? You know, different parts of the world? Is it that you find more of this or less?
Well, that's a good question. And we do find, for instance, there is some truth to that there is a group of Georgian Jewish people over in Israel who had a lot more of what we call more classical pan. Not as many strokes, and there seems to be more people in various places in Europe who might have some other manifestations, whether it's more of an immune deficiency type of picture. But it's really tough in the United States in general because we're a hodgepodge. We're a mix, right? So it can be easier over in some of those countries because there's just not quite as much mixing that goes on. But it does seem that there is, in some of these populations, more of a tendency for certain manifestations of the disease, although there's still some over there that had some strokes. It just wasn't as prominent, for instance, as ours has been.
Okay. Good. Last question.
Does an enlarged spleen have any correlation with diabetes?
Yes, I have a lot of data on the enlarged spleen that I talked about earlier. And what we find is that the enlarged spleen is seen in a lot of patients. And sometimes it's made worse by if you have liver involvement or might be at risk for developing liver problems, because there's a number of patients who in our group who have big spleens, no clinical liver problems, but when you plot it out, it looks using some scientific formulas, it puts them in the at-risk category for portal hypertension. Now, this was all done before we put them on medication, too. So, yes, the big spleen is very much a part of this.
Thank you both so much.